Now I'll go over it again. We just saw the contraction where the myosin head binds to the thin filament, undergoes a power stroke, and draws it closer. As long as this continues, the thin filament will be dragged along the thick filament, and the sarcomere width will decrease. And we're saying that this is explained by the sliding filament theory. And I use this as an example. If we look at this sarcomere, a relaxed sarcomere, it has the I band, which is the actin filament. It has the A band, which is the thick filament, myosin, right? And within the A band, we have the H zone, which is a lighter portion of the thick filament. And in the A zone, we don't have overlap between thick and thick filaments. Now, during contraction, if we started with a Z line here, that's our thick filament with the head sticking out. So the head, when it's activated, will bind to the thin filament here and undergo the power stroke by rotating. When it undergoes the power stroke, it draws the thin filament closer, or it draws the Z lines closer to each other. So this is the first power stroke. Then it detaches, binds to it, then power stroke, and draw the thin filament again. So you realize that the thin filament, or the Z line, is being drawn from this area to this area, close to the Z line. Now a similar thing will be happening here. So that at the end of contraction, we begin with a sarcomere of this width and ends up with a sarcomere with this width, with the H zone completely obliterated. It's completely obliterated. So in the contracted muscle, you don't have an H zone because of the sliding of the thin filament over the thick filament and the reduction of the Z line. Reduction of the sarcomere, the width of the sarcomere. Now, even though the width of the sarcomere is reduced, the length of the thick and thin filaments remain the same. Now, we talk about relaxation. We've seen excitation of the muscle. Now we're going to look at a contraction of the muscle. Now we're going to look at relaxation of the muscle. The muscle, when it contracts, would have to relax. Muscle do not contract interminably. They would have to relax. And relaxation begins when the concentration of calcium ions within the sarcoplasm decreases. Now, if the intracellular calcium ion concentration goes below 10 to the power negative 7 molar, then the relaxation will start. Because the calcium concentration has declined, the tropomyosin molecule or complex would change back into its original conformation. When it changes back into the original conformation, the tropomyosin filament goes to lie back in its original position, thus covering the myosin binding site of the actin filament. And this will result in the relaxation of the muscle because the myosin head can no longer bind to the actin filament because the binding sites have been covered Is it clear to this point? Decrease in intracellular calcium concentration results in the tropomyosin assuming its original conformation. This would ensure that the tropomyosin drops back into its original position and covers the myosin binding sites on actin. Once those sites are covered, myosin can no longer interact with actin and the muscle relaxes. Now we're going to look at the energy source for the muscle contraction. We've seen that both contraction and relaxation requires energy because the calcium reduction that we find during relaxation is due to the fact that calcium is actively pumped back into the sarcoplasmic reticulum. Now, active movement of particles from a region of low concentration to a region of high concentration, as we saw, requires energy. So both contraction and relaxation requires energy. Now we're going to look at the energy sources for this, the muscular activity. Now there are three sources of energy from the muscle. We have the creatine phosphate, glycogen, and cellular respiration in the mitochondria. Now, each of these processes would generate ATP for muscular activity, but the amount and the efficiency as well as the availability of the energy from these sources vary. Let's look at creatine phosphate. It's the ready source of energy in the muscle. Now, creatine phosphate will directly hydrolyze ADP. Now, the interaction between creatine phosphate and ADP is catalyzed by the enzyme creatine phosphoryltransferase. So you have creatine phosphate plus ADP giving creatine and ATP, and the ATP will be utilized for muscular activity. It's the most ready source of energy in the muscle. But the energy generated from creatine phosphate is low. It's not much, right? And this form of ATP generation or energy generation is called alactic anaerobic. It's called alactic anaerobic, or it's an alactic anaerobic source of energy. Alactic because no lactic acid is produced, and anaerobic because no oxygen is utilized. Now, this source of energy supply can sustain contraction for about just 5 to 8 seconds. So we have a creatine phosphate readily available. The creatine phosphate will phosphorylate ADP to get ATP, which can be utilized. Now, is this part clear? Our first source of energy supply. And this source of energy supply is short lasting, 5 to 8 seconds. Now, the second source of energy supply in the muscle is from stored glycogen. In the muscle, 1% of the skeletal muscle is glycogen, about 1%. Right. Now, glycogenolysis, the breakdown of glycogen, would produce glucose 1-phosphate, which enters the glycolytic pathway. Now, this generates pyruvate and ATP molecules. So, the ATP can be utilized. The ATP can be utilized, and the pyruvate, in the absence of oxygen, would yield acetyl-CoA, which will produce lactic acid. Now, talking about the energy sources for muscle activity, the immediate source of ATP is creatine phosphate. Now, creatine phosphate will phosphorylate ADP to produce ATP, and the reaction is hydrolyzed by the enzyme creatine phosphoryl transferase. Now, during this production of energy, no lactic acid is produced, and no oxygen is utilized. Now, this energy from creatine phosphate will last for between 5 to 8 seconds. So even though the immediate source of energy, it's not a very sustainable energy production process. Now, the next source of energy, now, Agnes, in the muscle, we have sarcoplasm, and within the sarcoplasm, we have lots of creatine phosphate. There are a lot of creatine phosphate. Now, this creatine phosphate would phosphorylate ADP. You know, ADP is a product of hydrolysis of ATP. If ATP is hydrolyzed, we get ADP and inorganic phosphate. This inorganic phosphate is usually used as the source of energy for most activity. So we have creatine phosphate. Now, this creatine phosphate will then phosphorylate the ADP. You know, ADP is not an energy producing molecule. It's ATP which produces the energy. So creatine phosphate will phosphorylate ADP to produce ATP. Now, this interaction is made faster by the enzyme creatine phosphoryl transferase. Now, the energy that is produced from this process is not much, and it's not sustainable. This energy will be depleted within 5 to 8 seconds, and it's the immediate source of energy supply in the muscle. Now, the next source of energy is from glycogen. In the muscle, we have stored glycogen. Now, glycogen is from glucose. When glucose is absorbed, it's stored mostly as glycogen. And about 1% of muscle is glycogen. So during activities, the glycogen is broken down, which is the process called glycogenolysis. Now, the next source of energy is glycogen. And I said about 1% of muscle is glycogen. The glycogen is broken down to produce glucose 1-phosphate, which goes into the glycolytic pathway. Now, this would always result in the production of pyruvate or pyruvic acid and ATP molecules. Now, the ATP molecules will then be utilized. Right. Now, in the absence of oxygen, pyruvate is converted into acetyl-CoA molecules. Now, these condense to form lactic acid. Right. Now, if energy is available, the acetyl-CoA is metabolized and goes through the Krebs cycle. Right. It is metabolized completely through the Krebs cycle. Now, the utilization of pyruvate during the anaerobic conditions would result in the formation of lactic acid. And this form of energy generation is called lactic anaerobic. It is lactic anaerobic because lactic acid is produced and no oxygen is utilized. We saw that the creatine phosphate is alactic anaerobic. Now this is because no lactic acid is produced, but oxygen is not utilized. Right? No lactic acid is produced, but and oxygen is not utilized. Now this source of supply of energy would easily be depleted within a short time. The energy produced would be used up, but it's longer than that of the creatine phosphate. The energy lasts within minutes instead of seconds, like in the creatine phosphate. Now, the third source of energy supply in the muscle is through the cellular respiration. Now, even though this process is slow, it's the most efficient of the three, and it will generate enough energy that would take longer time to exhaust. Right. Now, the substrate for this process is no, the substrates for these processes are fatty acids and glucose. Now, this will suffice for our purpose. If it were biochemistry, we'd have to go through the process. But for physiology, will be enough. So the energy sources will come from creatine phosphate, will come from glycolysis or glycogen, and will come from cellular respiration. Creatine phosphate is the most immediate, but not long lasting. The energy produced will be depleted in a few seconds. Glycogen will produce more energy than creatine phosphate, but this will be depleted in minutes. Cellular respiration takes longer to generate energy, but is the most efficient since it continuously generates energy and will be used for a long time. Now, during physical activity, all these three processes will be taking place, but the dominant one will depend on the time. Just when exercise starts, creatine phosphate will be producing most of the energy, but within a short time, the glycogen pathway will be producing most of the energy. But as the process goes on, then cellular respiration will be producing most of the energy. Now, this chart sort of summarizes what I've just said. The three energy sources, creatine phosphate. Creatine phosphate would phosphorylate ADP into ATP, which will be utilized by the cell. Right? And when it's utilized by the cell, we have ADP. Now, an enzyme catalyzes this process, the conversion of ADP to ATP by creatine phosphate. And they said the enzyme is creatine phosphoryltransferase. Now, this source of energy is not sustainable. Within seconds, it's exhausted. Then we have glycogen. Glucose gets into the cell, is stored as glycogen. Now, glycogen, when it's broken down in glycogenolysis, will produce ATP, glucose 1-phosphate, which goes through the Krebs cycle, right, to produce ATP and acetyl-CoA and pyruvate. Now, in the absence of oxygen, the pyruvate will be converted into acetyl-CoA, which will condense to form lactic acid. Now, the third source of energy is oxidative phosphorylation or cellular respiration. This is slow, but is the most efficient. It produces energy for a long time. So these are our three sources of energy production. Now, any questions on that? And these are the the way energy is produced. I mean, creatine phosphate will produce energy, and in no time, all energy is exhausted. Glycogen or glycolytic pathway will produce more energy and at a longer duration. Now, aerobic respiration, that's cellular respiration, or oxidative phosphorylation, will continue to produce energy for a very long time. I'll go over this again. We saw that contraction and relaxation of the muscle requires energy. ATP is used during the cross-bridge cycling. ATP is used to pump out calcium ions so that relaxation will take place. Now, it means that there should be a continuous supply of ATP in the muscle for contraction and relaxation. Now, three methods, or there are three sources for energy supply in the muscle. Now, these sources are creatine phosphate, glycolysis, and oxidative phosphorylation. I said there should be a continuous supply of ATP for muscular activity. And there are three sources of ATP in the muscle. First, creatine phosphate. Second, glycogen. And third, through oxidative phosphorylation or cellular respiration. Now, creatine phosphate is the immediate source of energy for the muscle. Now, this produces energy at a faster rate, but very little ATP is produced. And within 5 to 8 seconds, the energy produced from this process is depleted. Now, creatine phosphate will phosphorylate ADP to produce ATP. And this reaction is catalyzed by the enzyme creatine phosphoryl transferase. So it's the immediate source of energy for cell muscular activity. Then we have glycogen. About 1% of the muscle weight is glycogen. Now, during muscular activities, glycogen is broken down to glucose 1-phosphate, and this goes through the glycolytic pathway. When it goes through the glycolytic pathway, it results in the production of ATP. Right. It produces ATP, which is the energy source. Now, in the absence of in the absence of oxygen, the pyruvate will be converted into acetyl-CoA, which goes to produce lactic acid. Now, in the presence of oxygen, the pyruvate will be utilized, the acetyl-CoA will be utilized by going through the cellular respiration. Now, the third source of energy supply is by oxidative phosphorylation or cellular respiration. This process is slow, right, but is the most efficient because it produces lots of ATP that can be utilized. Now, the substrates for fatty acid, for oxidative phosphorylation are fatty acids and glucose. So these three processes would ensure that the muscle is supplied with energy. Now, at any one particular time during muscular activity, all three processes will be taking place. They'll be taking place, right? But the one that is predominant would depend on the duration of the exercise. And I said, for instance, if we look at this chart, at the onset of experiment, creatine phosphate will produce most of the energy. This is the energy, and that's the time. But within a short time, the energy produced will be depleted, comes to zero. Glycolytic pathway will produce energy slowly, but then within a few minutes, that pathway is not sustainable. Now, cellular respiration will begin producing slowly and will produce whilst the exercise lasts. This is the source of energy. Now, let's look at what happens when there is no energy. When ATP is depleted and creatine phosphate is also depleted in skeletal muscle, this results in a state of extreme rigidity called rigor. Know that you need ATP to separate or detach the myosin heads from the actin head. You need ATP to pump out the calcium ions. So when there is no ATP, the muscle will be in partial contraction, right? And this contraction, rigidity, when there is no ATP, is called rigor. In the death, this is called rigor mortis. That is, when this occurs after death, it's called rigor mortis. Good. Now let's look at the mechanical properties of skeletal muscle. Now, the skeletal muscle can generate tension in various ways. And there are two types of muscle contraction. We have isometric and isotonic contractions. Now, in isometric contraction, the muscle develops force, right? Even though it will be contracting at the same length, the muscle will be contracting, but the muscle will remain at the same length. It does not shorten. So that's why we call it isometric, contracting at the same length. Even though it's generating force, it does not shorten, right? And usually the force that is developed in isometric contraction is equal to the load on the muscle. An example of isometric contraction is trying to lift an unmovable object. If you try to move an unmovable object, then you are using isometric contraction. Right? So there is no movement. Even though the muscle is contracting, it remains at the same length. It remains at the same length and it exhibits no movement. Contraction at the same length. Even though tension is developed, the muscle does not shorten. Good. Now the next one is isotonic contraction. And in isotonic contraction, the muscle generates a constant force to move a load. A constant force is generated by the muscle to move a load. And there are two types of isotonic contraction. These are concentric and eccentric isotonic contraction. In concentric isotonic contraction, the muscle will shorten in length. Or the muscle shortens in length, and the force that is generated by the muscle is always less than the muscle's maximum. The force that is used during or generated during concentric isotonic contraction is less than the maximum force that the muscle can generate. And the contraction velocity is determined by the load. If you are lifting something and it's light, it's easier lifting it. But when it's heavy, it's slow. It's difficult lifting it, right? So we say the contraction with velocity. Now, example of concentric isotonic um, contraction is the lifting of weight, raising of a weight during a bicep curl, like this. See that the muscle is shortening. Yeah, the muscle is shortening, and this force generated is greater than the force on the muscle. That's why it can easily be lifted. Then we have eccentric and isotonic contraction. Here the muscle lengthens. It does not contract. It does not shorten. It lengthens. Even though energy is generated, force is generated, tension is generated, the muscle lengthens. And that's an example. Right, that's an example. The muscle contracts with force less than resistance and lengthens. And it's when we release the downward movement of the dumbbell in the bicep curl, the muscle lengthens instead of shortening. So in concentric isotonic, the muscle shortens, and in eccentric isotonic, the muscle lengthens. Now, we're talking about the muscle either staying the same length when working or shortening and lengthening. Now, the mechanical properties of a muscle is dependent on the contractile elements present in the muscle, and the contractile elements we've seen as the filaments, myofilaments, that would bring about the sliding filament theory. And the elastic elements, so contractile elements and elastic elements. Now within the muscle, in the muscle, we have contractile elements, those are the myofibrils, as well as elastic elements. The elastic elements are usually the some cytoskeleton, the tendons, and the connective tissue that we find around the cell. Now, for instance, in isometric contraction, the muscle length remains the same because the contract elements, even though are shortening, the elastic elements will stay same, so the muscle will look like it's not contracting. If you're depending on the muscle length, then you think it's not contracting, though power is being generated. Now, we talked about an eccentric contraction where the muscle lengthens. The muscle will lengthen because the elastic elements will stretch together with the contractile elements. So the mechanical properties that we've seen is dependent on the contractile elements as well as the elastic elements. Now we look at the muscle twitch. The muscle twitch. Now, we know the muscles in the body are innervated by motor neurons, right? And the motor neurons, when they are stimulated, will release transmitters that will cause the muscle to contract. Now, we saw that the neuron at the terminal end, it divides into the telodendria, or the terminal buttons. Now, these terminal buttons will innervate different muscle fibers, right? So the motor neuron and all the muscle fibers it innervates will constitute what we call the motor unit. A motor unit showing all the muscle fibers it innervates. So we have a motor unit. So I'll talk about the motor unit again. It's a motor neuron, and all the muscle fibers it innervates. We saw that in the neuron, the terminal end of the neuron will divide into terminal branches or telodendria. Now, each of these will innervate different muscle fibers. Right. So the motor neuron, together with the muscle fibers it innervates, constitutes what we call the motor unit. The force generated by the muscle depends on the motor unit. So the force generated would depend on the number of muscle fibers in the motor unit, the size of the individual muscle fibers, the length of the muscle fibers at rest, and the number of cross bridges that will be attached to the actin filament. So all these will determine the amount of force that will be generated by the motor neuron. Now, a twitch contraction is the quick shortening observed in a skeletal muscle when it's giving a single action potential. Right, when it's a single action potential is applied to the muscle, it will contract. That is, it will briefly become short. Now, this brief shortening of the muscle is what we call the muscle twitch. You apply a single action potential, and the muscle will shorten and come back to the original length. Now, this brief shortening observed in the skeletal muscle when an action potential, a single action potential is applied, is called the muscle twitch. If we take the motor unit, apply a single action potential, good, and the muscle shortening, the twitch has three phases. It has three phases. There is the latent phase. The latent phase, which is the duration between the stimulation of the muscle and the beginning of contraction, the time interval between the stimulation of the muscle and the beginning of contraction. And it's believed that this latent phase is the period where calcium ions will be mobilized from the sarcoplasmic reticulum. The second phase is the contraction phase, where the muscle will shorten. Then the third phase is the period where the muscle will get back to its original length, which is the the relaxation phase. Now, during the relaxation phase, calcium will be pumped back into the sarcoplasmic reticulum. During the contraction phase, there's a lot of calcium, and we have the formation of the cross linkages. That is the interaction between the cross bridge and the actin filament, which results in shortening or generation of the tension or force. So this is our twitch. Three phases: the latent phase, which is during the period during calcium mobilization; the contraction phase, that is the period during where the period where tension is developed; and the relaxation phase, where the period where the tension declines, and the periods where calcium is moved back into the sarcoplasmic reticulum. Now, we said that the motor unit is a neuron and all the muscle fibers that it innervates. Now, if a singular action potential is conducted in the motor neuron, it will cause a brief contraction in the muscle. Now, this brief contraction caused by a single action potential to a neuron is called a twitch. Is this clear? A single action potential to a motor neuron will cause a brief contraction of the muscle. Now, this brief contraction as a result of a single action potential to the motor neuron is called a twitch. It's called a twitch. Now, the twitch has three phases. It has a latent phase, which is during the period between stimulation of the muscle and the initiation of contraction. And it's believed that during the latent phase, calcium ions are mobilized from the sarcoplasmic reticulum into the sarcoplasm. Then we have the contraction phase. This is where force is generated, where the muscle becomes short somehow in some muscle, yeah? It's a period where we have increased formation of cross-linkages. Now the cross-linkages between the head of the myosin and the actin filament. So we have the generation of the tension. Then we have the relaxation phase, where the tension that is developed is eased. The muscle will return to its original length. And it's believed that during the relaxation phase, calcium is pumped back into the sarcoplasmic reticulum. We saw that during relaxation, the intracellular concentration of calcium drastically reduces. So this is our twitch. Agnesta, I hope you have understood this. Now, based on the twitch duration, now the twitch does not have same duration. The duration of twitch varies with the type of muscle that is being stimulated. Now, based on this twitch duration, there are three types of skeletal muscle. These are type 1, type 2A, and type 1B fibers. Now, the structures and biochemistry of these fibers are different. Let's look at the type 1 fibers. The type 1 fibers are also called the slow oxidative fibers. Now, these type of muscle fibers have lots of mitochondria. Now, they have lots of mitochondria because they utilize cellular respiration or oxidative phosphorylation to generate their ATP. So, there's lots of mitochondria. Now, these are resistant to fatigue. That is, they do not easily tire because they generate loads of energy. And they are rich in myoglobin. Myoglobin is a molecule, protein ion molecule that binds to oxygen. Now, because of myoglobin being present in high concentration, these fibers are red. Myoglobin is like hemoglobin, but hemoglobin in blood will bind to 4 molecules of oxygen, while myoglobin will bind to 1. Now, these molecules make the tissues red. Blood is red because of the presence of hemoglobin. Then, this type A or slow oxidative muscle have an extensive blood supply because they need to be supplied with blood which contains oxygen to ensure effective oxidative phosphorylation. Now, these are innervated by neurons which are small in diameter. So they are slow conducting neurons. And the type 1 or slow oxidative fibers are small and they are dominant in muscles responsible for posture. Then we have the type 2A or fast oxidative fibers. These are also red fibers. They are rich in mitochondria as well as myoglobin. They have extensive blood supply, just like in the type 1. And they are resistant to fatigue, but not as much as the type 1 fibers. And these are needed for sports such as middle distance running and swimming. So these will produce the energy would have energy for that. They do not tire air quickly because they generate lots of energy. Then we have the type 2 or fast fibers. Now these have few mitochondria compared to the type 2A and type 1 fibers because they do not really require they do not use oxidative phosphorylation as their source of energy. Rather, they use the glycogen because they are rich in glycogen. So they depend on the glycolytic pathway to generate ATP. And they easily fatigue. They easily become tired. They have low myoglobin, so they look white. They look whitish because the myoglobin level concentration is very low. They have large diameters and they are fast conducting motor neurons. They have fast conducting motor neurons. They have less extensive blood supply, of course, because they are not require any oxygen to generate energy. They have extensive sarcoplasmic reticulum. They are larger fibers, and they dominate in muscles used for rapid movement. Any muscle that is used for rapid movement would have predominantly type 2B fibers. Now, if you take most skeleton, they contain a mixture of these muscle fibers, type 1, type 2A, type 2B fibers. They contain all of them. But a single motor unit always contains one type of or the other, never all three. So if we take a single motor unit, it will contain only type 1 or only type 2A or only type 2B fibers. Now we look at summation and tetanus. Now, if we stimulate a muscle and let the muscle contract and stimulate it again, we'd have two twitches. First, we have our twitch, and we stimulate and we have two twitches. Now, if we stimulated a muscle and it's twitching, and give a second stimulus while contraction is in progress, the second stimulus together with the first will add up to generate a much larger tension. So you see, this is the first. It's still in progress when the second stimulus was applied. So the tension developed will be higher. Now, this condition where individual responses fuse together is called summation of waves. Summation of waves. Coming back here. So, if we give the stimulus while contraction is still in progress, or when the twitch is still in progress, the next contraction will produce a greater force, and this is called wave summation or summation of waves. Now, is it clear to this point? Well, that's our summation. The tension developed greater than the individual twitches. Now, if we increase the frequency of stimulation, like here, the response will be a singular smooth line. The individual responses will fuse to produce a singular smooth line like this. Now, this response, where the muscle is stimulated frequently without relaxation, is called a tetanus. So this here is a tetanus. Now we don't even see the individual responses. It's so smooth. All the responses are fused together because the frequency of stimulation increased. Now if we were to allow brief relaxation before application of the second stimulus, then we have this type of tetanus, which is called incomplete tetanus, because you realize there are some partial relaxations. So we have an incomplete tetanus. But when the frequency increases, these are the point of stimulation, all the individual responses will fuse into one to produce a tetanus. The tension developed by a tetanic contraction is about 4 times that developed by a single twitch. So we see incomplete tetanus and the complete tetanus. The tension developed by a complete tetanus is 4 times that developed by a single twitch. So we see our summation, and summation is due to the fact that the action potential of the muscle is much faster than the contractile response. So if we generate an action potential, a response will be generated. Now, because the action potential is over within a short time, if we applied another stimulus here, then we have summation of waves. We have the summation of waves. So coming back, so we have a twitch. If we applied a stimulus, we generate a response. And while the response is still in process, before the muscle relaxes, if we apply a second stimulus, then we have the response of the second stimulus fusing with the response of the first to produce what we call the summation of wave. Now, if we increase the frequency of stimulation, then the wave that becomes a single line with brief relaxation, that is. And this is called an incomplete tetanus. Now, if the stimulation rate increases further, then this will fuse into a single response, which is a complete tetanus. The tension developed in a complete tetanus is about 4 times that is generated in a single tetanus. Now, that will do with skeletal muscle. Now we're going to look at cardiac muscle. Now, the cardiac muscle has some features which are similar to the skeletal muscle. Now, both cardiac and skeletal muscle are striated, even though the striation in the cardiac muscle is not as pronounced as that in the skeletal muscle. Now, I was saying that there are certain features that the cardiac muscle has, which the skeletal muscle also have. Now, they both have striations, but the striations in the skeletal muscle is more pronounced compared to that of the cardiac muscle. Now, the cardiac muscle are found in the heart wall. They are responsible for the pumping action of the heart. So their function will pump blood from the heart to the other parts of the circulatory system. They are involuntary. because they are automatic. You can't regulate your heart, the activity of the heart, right? Because of that, they are involuntary. Unlike skeletal muscle, which you can tell, you can make a muscle contract. If I want my biceps to contract, I'll just do it. So it's involuntary. It's voluntary. Cardiac muscle is involuntary. Now, the cardiac muscle are branched, interlacing striated cells, right? They are branched. It's not like the skeletal muscle, which are cylindrical. So you see, that's cardiac muscle. The muscles are branched. But arrangement is similar to that of the skeletal muscle. Now, predominantly, cardiac muscle are mononucleate. For most of the cardiac muscle cells, there is only one nucleus. Skeletal muscle is multinucleate, and the muscle are found at the periphery. Now, they have the T-tubules, which are the tubes that invaginate from the sarcolemma into the cell. And we saw that the T-tubules are responsible for the conduction of action potential into the cell. They also have endoplasmic, sarcoplasmic reticulum, but that is not as prominent as that of the skeletal muscle. They are innervated by the autonomic nervous system, not the motor, the somatic system. So these are some characteristics of the skeletal muscle. Now the T-tubules in the cardiac muscle are found close to the Z line, and for each sarcomere there's a single dyad. Now the dyad is a interaction between the terminal system and the T-tubules. We saw that in the skeletal muscle, we have a T-tubule that is sandwiched between two terminal systems to form a triad. In cardiac muscle, we don't have a triad. We have a dyad because we don't have the T-tubules sandwiched between two terminal systems. We have the T-tubule lying close to a singular terminal system. And these two together will form a dyad. I said cardiac muscle cells are branched. They are mainly mononucleate. They have all the characteristics of the skeletal muscle. But unlike skeletal muscle, they don't have a triad. And I mentioned that the triad is made up of the terminal systems, the swollen ends of the sarcoplasmic reticulum, and sandwiching a T-tubule. The T-tubule is the invagination of the plasma lemma, that is the sarcolemma, and which gets into the cell and conducts action potential into the cell. Now, in the cardiac muscle, we don't have a triad. We don't have the T-tubule sandwiched between two terminal systems. There is a dyad. We have a T-tubule lying close to a single terminal system. So only two. So they form a dyad. Unlike in the skeletal muscle, where the T-tubule is sandwiched between two terminal systems to form the triad. Now, the cardiac muscle cells are innervated by the autonomic nervous system. They are innervated by the autonomic nervous system. Next, we look at the electrical activity of the cardiac muscle. The resting membrane potential of the cardiac muscle is about -90 millivolts. The action potential of the cardiac muscle is different from that of the neuron or the skeletal muscle. There are two general types. You have the non-pacemaker action potential and the pacemaker action potential. The non-pacemaker potential is like this. It has four phases, and these are what bring about the phases. Phase 0, that is the rapid depolarization, is due to rapid influx of sodium ions, just like in the neuron. Voltage-gated sodium channels will open and sodium will move into the cell, and this will cause the depolarization. Then we have an initial repolarization, where we would have chloride ions moving out and potassium ions moving out. Then we have this plateau, and during this plateau, there is calcium influx. And the calcium influx is balanced by potassium efflux. So we have this plateau. Then we have this third phase, which is the main repolarization. During this part, the calcium channels are closed and potassium are leaving in quantities. Even though a little potassium will be getting in, the repolarizing force is more than the depolarizing force, and this causes the repolarization to the original level. See that this is very different from the action potential of a neuron, which is similar to that of the skeletal muscle. So that is the non-pacemaker potential. Now in the heart wall, about 1% of cells are not muscle. They constitute what we call the conducting system. So about 99% of the heart wall is made up of the cardiac muscle. Just about 1% is made up of this conducting system. Now, this conducting system would generate action potential spontaneously. They are automatic cells. They don't need to be stimulated to generate an action potential. They generate action potential on their own. Now, the conducting system is made up of the sinoatrial node, which is the pacemaker of the heart, the internodal pathways, then the atrioventricular node, then the bundle of His, then the Purkinje fibers. These are the conducting system, and they will generate action potential by themselves. Now, the pacemaker generates action potentials at a fast rate, so it sort of overwhelms or regulates the others. Now, the pacemaker potential, when it generates the action potential, is conducted to every part of the heart wall by the other conducting systems. Then this will be conducted to the muscle fiber, which would cause contraction of the muscle. Now, because of this conducting system, if the heart is ripped out of the body and placed in a bowl, the heart will continue beating. It's because of this spontaneous generation of action potential by the conducting system, which is conducted to the cardiac muscle. Okay, I'll go over it again. I said that in the heart wall, about 90% of the material is cardiac muscle, which we've seen. There's 1% made up of what we call the conducting system. Now, this conducting system generates action potential spontaneously. They don't need to be stimulated to generate action potential. Now, the conducting system is made up of the sinoatrial node, the internodal pathway, the atrioventricular node, the bundle of hairs, and the Purkinje fibers. So the action potential is generated here, or could be generated here, but this one generates action potential faster and rapidly. So it's said to be the pacemaker of the heart. When the action potential is generated by the sinoatrial node, it is conducted to every part of the heart. Then through the atrioventricular node to the Purkinje fibers. Then the bundle of hairs to the Purkinje fibers, then to the cardiac muscle. So that this will generate action potential spontaneously and will be conducted to the cardiac muscle. And this will result in the generation of the cardiac action potential in the cardiac muscle. Now we'll continue. Now, this is the action potential of the conducting system, and this is superimposed on the action potential of the cardiac muscle, because this will generate that in the cardiac muscle. Now, the pacemaker, which is the sinoatrial node, is regulated by the autonomic nervous system. Now, sympathetic activation will cause an increase in pacemaker rate, and the parasympathetic activation will cause a decrease in pacemaker rate. That is why if you are involved in any activity which is under sympathetic control, your heart rate will go up. Under parasympathetic conditions, where you are relaxing, your heart rate could be normal or go lower. The sympathetic nervous system or the autonomic nervous system just moderates the activity of the conducting system, right? It moderates it. Now let's look at the contraction of the cardiac muscle. Now we've seen that excitation begins from the sinoatrial node. When this is activated, the action potential will be transmitted to the cardiac muscle, right? And this will cause contraction of the cardiac muscle. Now let's look at the excitation contraction coupling. When the action potential in the cardiac muscle has been generated, it will be passed on to the interior of the cell via the T-tubules. So this is our T-tubule. The action potential comes here. And in the T-tubule, we mentioned that we have the dihydropyridine receptors, right, which are calcium channels. Now, when the action potential gets to the calcium channels would open. They cause a change in conformation of the calcium channels, and this will open, and calcium will move out into the calcium will move into the sarcoplasm from the extracellular fluid. Now, the calcium that gets into the cell will cause the ryanodine receptors to open so that some more calcium will come out of the sarcoplasmic reticulum. Now, this process where calcium causes the release of calcium is called a calcium-induced calcium release. Calcium gets in there and causes the ryanodine receptors to open so that calcium will move out of the sarcoplasmic reticulum. So calcium is inducing the release of calcium from the sarcoplasmic reticulum, which is the calcium store. So we call this calcium-induced calcium release. Now, some more calcium will go into the cell via the calcium channels in the sarcoplasmic reticulum. Now, when calcium gets into the cell, it then causes contraction, just like in the skeletal muscle. But I want you to see the difference between how calcium is released from the sarcoplasmic reticulum in the cardiac muscle and that of the skeletal muscle. We saw that in the skeletal muscle, the dihydropyridine receptors are voltage sensors. They sense the differences in the voltage. So when the action potential is conducted in the T-tubule, the dihydropyridine receptors are activated. And this will activate the ryanodine receptors via the electron dense feet. And calcium will be released from the sarcoplasmic reticulum into the sarcoplasm through the ryanodine receptors. That is skeletal muscle. Now, in cardiac muscle, action potential will cause a conformational change in the voltage-gated calcium channels. Calcium will then get into the cell, and in the cell, calcium will activate. So, in the cell, calcium in the cell will activate the ryanodine receptors. The ryanodine receptor will release calcium. So here, calcium is inducing its own release from the sarcoplasmic reticulum. So it's said to be calcium-induced calcium release. Whilst in skeletal muscle, the dihydropyridine receptors will cause the opening of the ryanodine receptors to release calcium ions. Now, the calcium that is released, we know will bind to troponin C. Troponin complex will undergo a conformational change to shift the tropomyosin, just like in the skeletal muscle. And the myosin binding site on actin will be exposed. Next, we'll have our cross-bridge cycling, where ATP will bind to the myosin head. It's hydrolyzed into ADP and inorganic phosphates. This increases the affinity of the myosin head for actin. So it binds to actin, there's product release, the power stroke, and the drawing of the thin filament on the thick filament. Then next we have the binding of ATP to the head of the myosin, which causes detachment from the actin filament, and the cycle goes on, just like in the skeletal muscle. The difference between these two muscles is the calcium mobilization. One, in the skeletal muscle, we have dihydropyridine receptors causing the opening of ryanodine receptors by electron dense sheets to release the calcium. Whilst in the cardiac muscle, voltage changes will cause the calcium channels to open. There's influx of sodium ions. Sodium then causes the opening of calcium then causes the opening of the ryanodine receptors. And when these open, calcium will be released. Now, apart from this excitation contraction coupling differences, contraction in both skeletal and cardiac muscle are the same. Now, relaxation in the cardiac muscle is similar to that of skeletal muscle. Now, relaxation occurs when calcium is pumped back into the sarcoplasmic reticulum, or calcium is moved out through sodium-calcium antipodes. And you know what? We discussed an antipode. Now, during this process, this sodium-calcium antipode, 3 sodium ions will move in, and this carrier will pump out a calcium ion. Right, so it's an antipode. As a result of this active pumping of calcium ions, the muscle will relax because troponin will take its original shape. Tropomyosin will get into this groove and cover the myosin binding site, and relaxation will occur. Now, one thing I didn't mention or failed to mention is that the strength of contraction of the cardiac muscle depends on the concentration of calcium ions in the sarcoplasm. So the higher the concentration of calcium in the cell, the greater the strength of contraction. Now, let's look at the mechanical properties of the cardiac muscle. Now, the action potential of a cardiac muscle is almost the same duration as the mechanical response. That's the mechanical response, and that's the action potential. Now, the duration is almost the same. Now, because of this fact, or because of this, cardiac muscles do not tetanize. There's no tetanization in cardiac muscle. There's no summation of wave in cardiac muscle, like we just saw in the skeletal muscle, where when the muscle is still contracting, you can stimulate it and a second response can be generated. And these two will fuse to produce a much greater response. That's in summation of waves. In cardiac muscle, there is no summation of wave because the action potential and the mechanical response are almost same in duration. And we know that when the action potential is in progress and you apply a second stimulus, it will hardly respond to the second stimulus. And because of this, in cardiac muscle, there is no tetanus. Now we're looking at smooth muscle. The smooth muscle, usually these are in a form of sheet and they line the walls of hollow organs. Now, smooth muscle cells are spindle in shape. Spindle in shape, and they have a singularly placed or single-placed nucleus. So that's a typical structure of the smooth muscle cell. Now, the smooth muscle cell have the contractile elements they have the thin and thick filaments. Now, the thin and thick filaments attach to what we call dense bodies. Dense bodies which are found in the sarcolemma or in the sarcoplasm. So we have dense bodies to which the myofilaments are attached. Now, the dense bodies are analogous to the Z lines that we saw in skeletal muscle and, of course, in cardiac muscle. Right, but these Z lines are found in the muscle, and these can be in the dense bodies are found in the membrane as well as in the sarcoplasm. Now, in addition to the contractile element, that is the thin and thick filament, there are these non-contractile filaments called intermediate filaments, which aid in the shortening of the smooth muscle when they are activated. Now, the smooth muscle cells on their surface have these many flask-like invaginations, and these are called caveoli, the caveoli, and they are believed to um, to ferry calcium ions from without the cell into the cell. Now, the individual cardiac muscle fibers have gap junctions. Now, these are areas where electrical activity and substances can easily be passed on from one cell to the other. So the gap junctions allows chemical communication as well as electrical communication. Now, there are two types of smooth muscle. We have the visceral or unitary smooth muscle, and we have the multi-unit smooth muscle. Now, the visceral smooth muscle is made up of fibers bound together to form a single unit. So, we have a lot of muscle fibers, individual smooth muscle fibers bound together to form a single unit. And these will line the walls of hollow tubes. Now, these cells behave as a single cell because they have gap junctions which will transmit action potentials or chemicals from one cell to the rest of the cells. So if action potential is generated in this cell, the information will be transmitted to all these cells, and these will contract simultaneously. Now, the single or unitary visceral smooth muscle can generate action potential spontaneously. They generate action potential spontaneously, and they are lowly innervated. That means there are very little innervation, and they behave as a syncytium, behave as a single cell. So they exhibit plasticity and they can contract when stretched. Now, this means that when they stretch out or tension develops, the tension will ease easily, right? Let's say, for instance, the uterus. As the fetus grows, tension will develop, but these smooth muscles do not have length-tension relationship, right? So the tension that is developed will ease to a point where it's even much lower than the one that developed. So they exhibit plasticity. Now, the multi-unit smooth muscle, which is the second smooth muscle, are made up of discrete individual fibers, right? And they are highly innervated, and they have a resting membrane potential. They don't need to generate an action potential to generate a response. The least stimulation will cause them to contract, right? And they line large vessels. No, the multicellular smooth muscle are found in small blood vessels, while digestive tract, urinary system, and reproductive system, while the multi units are found in the large blood vessels, they will be found in the ciliary body, and they found at the base of hair follicles. Now they have high innervation density. They do not have interconnecting bridges, like they don't have these gap junctions that would allow electrical activity to move from one cell to the other. The membrane potential is stable. They cannot generate action potential spontaneously, and they do not display action potentials when stimulated to contract. So this is the difference between the multi-unit smooth muscle and the unitary or visceral smooth muscle. There is no interconnection. Here there is interconnection. The innervation density low here, innervation density high here. Right. So these are the differences mainly. Apart from that, their contraction would go in. And of course, we said that these will generate action potential spontaneously. These have a stable resting potential. Now, this is the slow wave, and is a means by which the unitary smooth muscle generate action potentials spontaneously. There is an alternating band of depolarization and repolarization. Now, when the depolarization reaches a threshold, then an action potential is generated in spikes. Then the slow waves will begin again. Now, as a result of this activity, the smooth muscle can generate action potential spontaneously. Now, let's look at the contraction of the smooth muscle. Contraction of the smooth muscle. Now, smooth muscle contraction is usually initiated by agents that will increase intracellular calcium concentration. The smooth muscle has low or a few voltage-gated sodium channels compared to the other excitable tissues. Flow of calcium into the cell is the main is a means by which the smooth muscle becomes depolarized. Unlike in the other excitable tissues where depolarization is due to influx of calcium ions, in smooth muscle is due to the influx of calcium. In the other excitable tissues, the depolarization is due to influx of sodium ions. In smooth muscle, there's going to be increase in calcium ions for depolarization. Now, the intracellular calcium ions can be increased in three ways. This is by opening of voltage-gated calcium channels. They are voltage-gated channels. These were open when the voltage changes. So action potential will cause opening of the voltage-gated calcium channels or voltage-operated calcium channels. Then we have the ligand-operated calcium channels. Now these ligand-operated calcium channels normally will bind to a messenger. A messenger will bind to the receptor, and when it does that, this channel will open and there will be influx of calcium ions. Now, there is another way by which calcium can be mobilized to. This, and we have the calcium-induced calcium release. So when voltage-operated calcium channels open, calcium gets into the cell, then calcium would cause the opening of receptors, which are IP3 receptors. And these receptors, when they open in the sarcoplasmic reticulum, calcium will flow into the sarcoplasm. Now, another way by which intracellular calcium can be increased is by ligands binding to receptors. And we've seen this, a ligand, external particle, a hormone, anything. This will bind to the channel. When it binds to it, it's a conformational change which will allow influx of calcium ions. Now, another way calcium can be mobilized is by using the IP3 pathway. Here, a substance will bind to a channel. The channel activates a G protein, which in turn activates phospholipase C, an enzyme. Now, phospholipase C will catalyze the conversion of PIP2 to IP3, inositol trisphosphate. Now, inositol trisphosphate together with DAG, diacylglycerol, would cause the sarcoplasmic reticulum to release calcium into the sarcoplasm, right? So these methods would cause an influx of calcium ions in the sarcoplasmic reticulum, in the sarcoplasm. I'll go over it again. Now, unlike the other excitable tissues that we've seen, depolarization in the cardiac muscle is dependent on calcium influx rather than sodium influx. Depolarization is due to calcium influx rather than sodium influx. And there are various methods by which calcium gets into the sarcoplasm. First, we have the voltage-operated calcium channels, or voltage-gated calcium channels. So that unlike the other excitable tissues where depolarization is due to sodium influx, in smooth muscle, depolarization is due to calcium influx. Now, there are various ways by which the intracellular calcium concentration can increase in the smooth muscle. First, by utilizing the voltage-operated calcium channels. Two, by using the ligand-operated calcium channel. And three, by calcium-induced calcium release. So now we look at what these are. Now, changes in the membrane potential will cause the opening of voltage-operated calcium channels. Now, these voltage-operated calcium channels would open and calcium gets into the cell from the extracellular fluid. So that's by way of the voltage-operated calcium channels. Now, calcium, when it gets into the cell, can induce release of calcium from the sarcoplasmic reticulum, and that is the calcium-induced calcium release. So this is the voltage-operated and calcium-induced calcium release. Now let's look at the ligand-operated calcium channels. Now here, an external factor such as a drug, such as a hormone, will bind to a calcium channel. When it binds to this, the channel will open to cause influx of calcium ions. So this is one method by which calcium gets in through the ligand-operated calcium channel. Then calcium can also get in through another method. This is by using second messengers. Here, The calcium binds to the receptor. Calcium binds to a receptor, and the complex that the ligand binding to a receptor, the ligand receptor complex will activate an enzyme called phospholipase C via a G protein. Phospholipase C will convert PIP2 to IP3. IP3 is a second messenger. Now, the PIP2 is converted into IP3 and DAG, which are both second messengers. IP3 will go to the sarcoplasmic reticulum, where it will bind to IP3 receptors and cause release of calcium into the cell. Now, the calcium together with DAG will move to the membrane channels and cause opening of these channels. So as a result of these activities, the intracellular calcium level goes up. So now let's go to contraction. Calcium contraction. Now in the smooth muscle, there is troponin, there is no troponin in smooth muscle. Tropomyosin and actin. There is no troponin, so take note of that. Now the calcium that gets into the cell will bind to a molecule called calmodulin to form a calcium calmodulin complex. Now the calcium calmodulin complex will activate a protein called caldesmon. Activates a protein called caldesmon. Now the caldesmon will then cause the shifting of tropomyosin in the actin filament to expose the myosin binding sites. Some calcium calmodulin complex will activate an enzyme, myosin light chain kinase. Now the myosin light chain kinase will then activate myosin light chain in the myosin. Once the myosin light chain is activated through phosphorylation, then cross-bridge cycling would begin. Right. So unlike the other two where calcium will bind to troponin C, and this will cause movement of tropomyosin and cross-bridge continuum. In the smooth muscle, calcium will first bind to its molecule calmodulin, 4 calcium ions to a molecule of calmodulin. The calcium calmodulin complex is an enzyme, and this will activate caldesmon. Caldesmon, when it's activated, will cause the shifting of tropomyosin from its original position to expose the myosin binding sites. Now, calcium calmodulin will also activate a kinase, an enzyme called myosin light chain kinase. The myosin light chain kinase would then activate myosin light chain through phosphorylation. So it utilizes ATP to activate the myosin light chain. The myosin light chain, when it's activated, will go through the cross-bridge cycling. It will hydrolyze ATP to form ADP and inorganic phosphate. Now, this will then bind to the thin filament, and the cross bridge will cycling will go on just like in skeletal and cardiac muscle. Now, in smooth muscle, as did I make sense to this point? I don't want to continue before we go asking questions. We saw that calcium concentration, intracellular calcium concentration goes up in the smooth muscle when they are activated. Now the calcium that gets into the cell will bind to a protein which is purposely for calcium binding called calmodulin. So the calcium will bind to calmodulin to form a calcium calmodulin complex. Now the calcium calmodulin complex would activate a protein called caldesmon. Now caldesmon, when it's activated, will cause the myosin filament to shift off its original position. And this will expose the myosin binding sites on actin. Good. Now some of the calcium calmodulin, which is the second messenger, we have a lot of them, will then activate a kinase, an enzyme called myosin light chain kinase, MLCK. Now, the myosin light chain kinase, when it's activated, will activate myosin light chain, an enzyme in the thick filament. Now, is it clear to this point? Is it clear to this point, please? Good. The myosin light chain, when it's activated, would make the cross bridge activated. You see here, the cross bridge needs to be activated before it goes to the cross bridge cycling. So once it's activated, ATP will bind to it. ATP will be hydrolyzed into ADP, inorganic phosphate. And we have our power stroke, then contraction, then detachment, just like we saw in skeletal and cardiac muscle. Now, before the cycling, these processes I just described had to take place. And I told you that smooth muscle has only actin and tropomyosin in their thin filament. There's no troponin. The work of troponin, you can see, is taken over by calmodulin. Now, is it clear to this point? Is it clear to this point, please? Good. So, the muscle will contract going through the cross-bridge cycling. Now, when calcium concentration falls in smooth muscle at a certain concentration, the smooth muscle, when it falls in the smooth muscle, the myosin light chain becomes dephosphorylated. It becomes dephosphorylated when the calcium level falls. Now, this dephosphorylation is by an enzyme called myosin light chain phosphatase. The myosin light chain phosphatase dephosphorylates the myosin light chain. Now, when that takes place, the activity of the ATPase on the myosin head is compromised. So the myosin head will not react with ATP and it wouldn't detach from the actin filament. Now, because it stays attached to the actin filament, there's still tension developed. And this mechanism is called the latch bridge mechanism. Now, this tension allows smooth muscle to maintain tone. It allows smooth muscle to maintain tone with a minimum expenditure of energy, patience. So it made sense. So even though the concentration of calcium has declined, tension is still developed, and this tension uses very little energy, right? So the latch bridge mechanism will ensure that tone is still maintained in the smooth muscle. Now let's look at the relaxation of smooth muscle. Now, relaxation involves actively pumping out of calcium ions. Calcium will move from the sarcoplasm into the sarcoplasmic reticulum using a calcium ATPase. And calcium will move out from the sarcoplasm to the extracellular fluid using a calcium ATPase or a sodium calcium exchanger, which I described not long ago. And when the calcium levels fall below a certain level, then the muscle will relax. So that's a relaxed, smooth muscle here, and that's the contracted, smooth muscle with all the contractile filaments. Yet this relaxed and the contracted. Now, length-tension relationship. Smooth muscle does not have any length-tension relationship. Cardiac muscle and skeletal muscle have this relationship. Now, if the length of the cardiac muscle, within limits, if the cardiac muscle is stretched and maintained at a certain length, tension develops in that length. Now, if you stretch it further, another tension will develop. If you stretch it further, same will happen. So that you can plot a graph of tension against length. You know that at this length, the tension is like this. Now, this exists in the cardiac and skeletal muscle. This relationship does not exist in smooth muscle. Now, if smooth muscle is stretched to a certain limit and length, tension develops, but the tension eases out. It eases off, sorry. So you cannot have a relationship between length and tension, unlike that of smooth muscle. Now, this is very significant in the function of smooth muscle, so that either bladder or uterus, when tension develops, this tension decays, so we can decide to do some activity. Let's say the bladder, for instance. At times you feel like going to the washroom, but you decided you are not going again. Then the tension will ease. Now you don't feel it. But at a certain point, there's no way you can decide that you won't go. Because the second characteristic of smooth muscle will be manifested, which is as smooth muscle stretches, it contracts. Now, so within limits, this is what happens. That ends your lecture on general physiology.